Yeah, very bright.
Hi, welcome to Science Hangout from Science Mission. So we have Dr. Kalina Hristova with us from Johns Hopkins University. She is the professor in material science and engineering department. She has won several awards. She has in 1988 won the Gold Badge of Scientific Merit from the University of Sofia. She won in 2000 Career Development Award from UC Irvine. She won the Margaret Oakley Day Off Award from Biophysical Society. She also won Marlin U. Zimmerman Faculty Scholar, Whiting School of Engineering currently. She was the chair in 2012 Membrane Structure and Assembly Conference. She was also chair in 2012 FACEB Research Conferences. She was elected to counsel the Biophysical Society. In addition, she has published several outstanding research in various publications. She is also in the various editorial boards and evaluated various publications for several journals. Welcome, Dr. Hristova.
Thank you for having me.
Nice having you on a Science Hangout. Can you please tell about your background, your education, and some of the work you are doing in your lab?
Okay, so I have a bachelor's degree in physics from the University of Sofia in Bulgaria. I have a PhD in mechanical engineering and material science from Duke University. Then I did a postdoc at the University of California at Irvine in the departments of physiology and biophysics and in pathology. And then since 2001, I moved to Hopkins, where I started as an assistant professor. So I am now a professor in the Department of Material Science and Engineering. Also, I'm a member of the Institute for Nanobiotechnology and a member of the graduate program in molecular biophysics here at Hopkins.
Okay. And like when you deciding your career, why did you think about academics compared to the industry? So is there anything which you thought helped you in academics, or like what was the reason behind your move to the academics?
Well, I don't know much about life outside academia, but I honestly cannot imagine that is as exciting as doing research in a university setting. And this is because we are really free to pursue the questions that really interest us, that tickle our imaginations. So the big questions of the day as we see them.
Okay, that is interesting. And do you have any mentors who supported you in your career?
Of course, we get a lot of support. So I've been supported and mentored by my teachers since grade school up to now. So for instance, now at Hopkins, I interact a lot with Michael Edidin, who is in the biology department. So he was one of the first people to discover lateral mobility in plasma membranes. So he made this discovery in 1970, and he's really a giant in the field, but he still finds time to read the dissertations of my students, to edit them and find typos and discuss the science with us and help us move forward. So I consider him my mentor and role model.
Okay, good. And what are the challenges do you face every day other than the research part? Like you manage the grants and the grad students, postdocs. So what are the challenges do you have every day?
Well, it's difficult. It's difficult to be a professor at a research university, and some of the challenges come from the insufficient funding. So, for instance, in our case, we work with very expensive equipment. We have very fancy microscopes.
Yeah.
So if something like this fails, something that is so expensive, it would be very hard to replace it because of the lack of funds. But at the same time, we cannot produce without this equipment.
Yeah.
So this is a challenge, you know, with that is faced by people that use, you know, state-of-the-art equipment.
Okay. And you are busy, I know, like throughout the day with your work. So if you find some spare time, how do you spend it? Like, what are your hobbies and what else do you do during weekends?
So I don't have spare time. I work during the weekends, but I have to say I do love to travel. So actually, I have to travel a lot for my job to go to meetings or to universities and visit colleagues. So for some people, that would be a burden. But for me, it really isn't, since I enjoy it. So I enjoy seeing new places.
Okay, that is nice. And can you please talk about your lab and lab people?
Sure. So my lab has 7 graduate students and 1 postdoc. And they come from different backgrounds. So 3 of my students are going to get a degree in material science in engineering. 4 of them are going to get a degree in biophysics.
Okay.
And also, they come from very different backgrounds. Some of them have chemistry backgrounds, some of them have physics backgrounds, some of them have biomedical engineering backgrounds, some of them have chemical engineering backgrounds. So really, we are a team of people, you know, with many backgrounds, students with many backgrounds. So we think of ourselves as physicists, engineers, and biologists, and all that.
Okay. And for these grad students, postdoc, usually like it takes long time to get their PhD, or like they do several years of postdoc. How do you keep them motivated every day to make sure that they are on top of everything?
I don't think they need much motivation. They are very motivated. I think they are more motivated. So what we do is we talk about the big questions, and I think they have a real desire to pursue the answer.
Okay. Okay. And like I saw some recent work from your lab, which is very much interesting. So can you please go ahead and present your work to everybody?
Sure. So I will give a short research presentation. So the thing about, before I start, let me just tell you that in our lab, we are interested in processes in biological membranes. And we have 2 sets of projects. So the first set of projects focus on interactions of proteins in biological membranes. So we are particularly interested in receptor tyrosine kinases because the activity of these receptors is controlled via their lateral interactions. So we develop methods to study this. But then we have a second set of projects that I'm not going to talk about today, which focus excuse me, which focus on interactions of small peptides with membranes. So these are called membrane active peptides. And we are interested in learning how to breach the plasma membrane or even breach endothelial layers with such peptides, so we can use them for drug delivery applications. So this is what we do. And now I will see if I can start my presentation. Go at the beginning. So do you see it?
Yeah, go ahead.
Okay. So today I will be talking about these receptors called receptor tyrosine kinases. So this is a very large family of receptors. There are 58 members organized in 20 different families. So as you can see here, they are single pass membrane receptors. They have an extracellular ligand binding domain. So this is the domain that binds the ligand, which is the green molecule, a single transmembrane domain, and a tyrosine kinase domain inside the cell. So this is an enzyme. So these receptors can form dimers, and it turns out dimerization is absolutely required for their activity. And this is because a tyrosine kinase in isolation cannot activate itself. But if they are brought together, they cross-phosphorylate each other on selected tyrosine residues on their activation loops. And this activates them. So now that they are active, they phosphorylate additional tyrosine residues on the receptor, and also they phosphorylate soluble proteins. And they bind adapter proteins, and they trigger signaling cascades that propagate from the cell membrane to the nucleus. And the results of these signaling cascades can be cell growth, differentiation, motility. So these receptors are very important for the life of the cell. They're also very important for human development, and they play profound roles in human disease, particularly in developmental abnormalities and in cancer. So for many years, these receptors were believed to dimerize only in response to ligand. So ligand is considered absolutely necessary for the dimerization because the receptors were thought to not have a propensity to interact, that ligand is needed to bind them, so it induces their ability to interact. But recently it has become clear, and this is through work from our lab and other labs, that the mode of activation is in fact much more complex, that these receptors can also form these unliganded dimers. So they do have an intrinsic propensity to interact. So these unliganded dimers form, and they are activated to a low degree, and then ligand binds, and then these unliganded dimers transition to liganded dimers with different structures and with higher activity. So one thing that we found is that for some receptors, the binding of different ligands results in a different structure and different activity, and this is the mechanism through which different ligands are recognized. Another thing that we found is that actually this intermediate, that unliganded dimer, is very important also, because if you perturb it somehow, then the ligand that binds cannot help the transition to the fully active state. So that fully active state is never achieved. So it appears that these unliganded dimers are important intermediates. And we see all this because in our lab, we developed methods to study the interactions between these receptors. So by now, you know that interactions are critical for function, but it is very difficult to quantify them in the plasma membrane. So what we do is we use Forster resonance energy transfer to characterize these interactions in quantitative terms. So what we do is we attach fluorescent proteins. So these fluorescent proteins have to be a FRET pair, which means that the emission of the donor has to coincide with the excitation of the acceptor. And when these receptors are monomeric, if you excite the donor will emit. So in this case, what I've drawn is M turquoise, which is a good donor for us. So all you will see is the emission of the M turquoise. But if a dimer forms, then when you excite the donor, the energy will get transferred in a non-radiative way. And then the acceptor will emit. So we will capture this emission. So the idea of FRET is very simple, but it is very tricky to turn it into a quantitative assay. So here I've given some publications that address this issue. So practically what we've done is we've developed the methods that allow us to determine the value of the equilibrium constant, the association constant, which is a fundamental physical chemical characteristics that describes the strength of the interactions in quantitative terms. So FRET is widely used, but in fact it is pretty tricky to use it because FRET is not exactly an assay for interactions, because what we measure depends on the value of the equilibrium constant. but also it depends on the so-called intrinsic FRET, which does not depend on the dimerization. It depends only on the structure of the dimer that is formed, and in particularly on the distance between the fluorescent proteins in our system, because we have proteins attached on flexible linkers. So here is an equation that relates intrinsic FRET to distance between the fluorescent proteins. It is written under the assumption of free rotation of the fluorescent proteins. So what we do in our work, we determine the intrinsic FRET, and there are no assumptions there. And then you can infer what the distance is. But in fact, the value that we use really is the measured intrinsic FRET. And we can assess if structural changes occur, let's say, upon ligand binding, but by looking at this value of intrinsic FRET. So before I talk more about FRET and about receptors, just let's look at an image of a cell which has been transfected with a receptor of your choice tagged with a fluorescent protein. So these receptors are made in the ER. They're folded there, and the fluorescent protein is folded there. So the construct becomes fluorescent in the ER, and that it is moved through the Golgi to reach the plasma membrane. So this is why you see it inside the cell pretty much everywhere. Also, the receptor is downregulated. So it is uptaken via endocytosis. So then you would see it also in the endocytic vesicles, and that's why you see this diffuse fluorescence. Furthermore, if you look at the plasma membrane, what you're going to see is, pretty impressive heterogeneity. So just by looking at images like this, you understand that in order to make precise measurements, you need to be able to somehow measure FRET and also concentrations for our applications over a very small area, a small membrane patch, or even better, every pixel of an image. So in our lab, we have a microscope like this. So this microscope was developed by our collaborator, Valerica Raico. So he's at the University of Wisconsin in Milwaukee. It's a 2-photon microscope, and what's unique about it is the optimist detection system. So the light that comes out of the sample is passed through a transmission grating. As a result, the light is decomposed into its entire spectrum. So this is a microscope that gives you spectral information in each pixel of the image. So here I've blown up an image of a cell so you can see the different pixels. And in each and every pixel, we get a complete spectrum. And it's very useful to have complete spectra because you know what the underlying spectra of the donors and acceptors are. So you can unmix the two and you can determine FRET efficiencies with very high precision. So you need FRET efficiencies, but then it turns out you also need the donor concentration and the acceptor concentration, so you can add them up and get the total receptor concentration, which actually drives dimerization. So in our lab, we have developed a method that can give you this with very high precision. So the method was developed by Chris King, who is a graduate student. He is a member of the biophysics graduate program here at Hopkins. It is called Fully Quantified Spectral Imaging. And it gives you the 3 parameters, the FRET efficiency, donor concentration, and acceptor concentrations without any assumptions. And what you need to do is you need to perform 2 scans of your samples. First, you have to excite the donor, and then you'll see the emission of the donor and the sensitized acceptor emission. And then you will excite now the acceptor directly, and you will see the emission of the acceptor. And I'm not going to go through these equations. They seem scary, but they're really not. They're very simple, but they're precise. And if you are interested in studying interactions, even soluble protein interactions or protein DNA interactions, these kind of equations can be very useful to you. And here is the citation. The paper was just published and we'll be very happy to answer questions about it. So furthermore, Chris has implemented this in a program that has a very friendly user interface. So everybody can use it. So we have a lot of undergrads that use this program and help us with data processing. So the idea then of these experiments is you co-transfect cells with genes that encode for your receptor of interest labeled with donor and with acceptor. So it's a co-transfection. And then the cell will make the protein for you. It will fully glycosylate it. It will export it to the plasma membrane. And then you subject the cell to FSI imaging and you determine these 3 parameters. So you determine concentrations and FRET efficiencies. Okay, so then we discovered something quite interesting. I already mentioned to you that just by looking at the membrane, you see a lot of heterogeneity. And here, when we determine the effective concentrations of receptor per pixel, again, we can see sometimes a lot of heterogeneity. You see here is this hotspot of high concentration. But one thing we can do better than average is we also measure the FRET efficiency with very high precision. And what we see is that the FRET efficiencies per pixel are actually pretty homogeneous. It's pretty much the same FRET efficiency. And this is quite peculiar because if this is indeed the concentration and this is the FRET efficiency, that would violate the law of mass action. So this is a fundamental physical chemical law that says when I have more receptors, I have to have more dimers. But then if I have more dimers, I have to have high FRET efficiency. So that law cannot be violated. So this actually gives us the idea that what we are looking at here is not really related to the real 2-dimensional concentrations of the receptors in the plasma membrane. That is an artifact. And in fact, recent work has suggested that the plasma membranes of cells are extremely wrinkled, that the cells possess 2 to 3 times the surface area needed to sustain their shape. And probably what we are looking at are the twists and turns of the membrane as it passes multiple times through the voxel of the image. So how do we prove that? Well, one way to prove that is to actually stretch the membrane. So it turns out you can apply reversible osmotic stress, which means that you can swell the cell without killing it. And once you put it back in normal medium, that it will recover and live happily ever after. But it will stretch the membrane. And what we see is that FRET efficiency does not change upon stretching. So what this is telling you is that the interactions are not changed upon stretching. All we are doing is we are unwrinkling the membrane. And and then you know the topology of your membrane perfectly. In this case, the membrane is perpendicular to the point of view. We're slicing through the membrane. It is very thin, so it's a diffraction limited structure. And the FRET efficiency are the FRET efficiencies are as homogeneous as the total concentration. So bottom line is, if you want to make quantitative measurements of association constants in the plasma membrane, you need to stretch the membrane. You are not going to kill the cell, so these will be still measurements in live cells, but you need to perturb it. So in our lab, we make measurements in live cells under reversible osmotic stress, or we have a different model system, which is a very beautiful model system. It's called plasma membrane-derived vesicles. So you stress the cells by subjecting them to a buffer with high salt that doesn't exactly kill them, but makes them apoptotic. So as a reaction, they start blebbing. They give us their membrane in the form of these beautiful vesicles. And we have developed protocols to produce these vesicles without the use of harsh chemicals such as formaldehyde or DDT. Because protocols in the literature actually have formaldehyde, and we've shown that the presence of formaldehyde introduces artifacts in our measurements.
Okay.
So these are alternatives to the swollen cells, and in fact, we make measurements in both systems and we see the same interactions.
Mm-hmm.
So now that I have talked about methodology, I am going to tell you a brief story about one of the receptors. So I chose this particular project because it is actually the shortest story. So I will tell you about something about the FA2 receptor. So the FA2 receptor belongs to the largest family of receptor tyrosine kinases called the F receptors. And here is its architecture. So here is the transmembrane domain. And these are the extracellular domains. And at the end is the ligand binding domains. And inside the cell, there is the tyrosine kinase domain. Then there is a SAM domain and a PDZ domain. So this work with the F receptors is done in collaboration with Elena Pasquale. So she's all the way in San Diego, but that does not stop us from having a very effective collaboration. So based on work that we've done, we are thinking of this architecture as follows. The really important domain is the tyrosine kinase domain. This is the domain that mediates activity. But all these other domains actually control interactions. So because interactions regulates activity. So you can think of them as regulatory domains. So the F receptors are very interesting, and one reason is that their ligands are not soluble factors. They are anchored to the plasma membrane together with the receptors. So what happens when two cells come together, the receptors from one cell and the ligands from another cell interact and co-cluster. So then these very large clusters are formed, and the tyrosine kinases get phosphorylated. So for these receptors, dimerization is not enough. They form large clusters. And then when the tyrosine kinase domains are phosphorylated, then they trigger signaling cascades that generally lead to inhibition of cell migration. So as you know, cell migration generally correlates with invasiveness and metastasis. So because of this, the inhibition of cell migration can be considered anti-tumorigenic signaling. So these receptors in the presence of ligand mediate anti-tumorigenic signaling. But something strange happens in the absence of ligand. In the absence of ligand, these large clusters do not form since it's a co-clustering event. But the signaling turns pro-tumorigenic. So this is very peculiar for a receptor tyrosine kinase. And in fact, there are cancers where people find the overexpression of the receptor and the loss of the ligand. And furthermore, these cells that don't have ligand seem to move faster, suggesting this pro-tumorigenic signaling. So we were wondering whether these receptors form dimers, just like all the other RTKs that we worked with, because it had not been reported before. So we decided to investigate that question. So what we did is we created two plasmids. One encodes for the receptor that is labeled with the donor via a 15 amino acids, like a flexible linker. And then the other one encoded for the very same receptor, but now the labeling was done with the acceptor, again, via flexible linker. And then we co-transfected the cells. So these are HEC-293 cells with these receptors. And when the receptors were made, glycosylated, and sent to the plasma membrane, Then we subjected the cells to reversible osmotic stress. And actually, we analyzed small portions of the membrane of the cells just to make sure that that this region is homogeneous and also its topology is very clear, that is perpendicular to the field of view. And we determined 3 parameters: donor concentration, acceptor concentration, and FRET efficiency per each small area chosen like this. And then we analyzed hundreds of cells. In fact, between 200 to 500 cells are being analyzed for each single construct in our experiments.
Wow.
So this is what data looks like. So as I said, we determined 3 parameters: FRET efficiency, donor concentration, and acceptor concentration. So what I do here, I plot them this way: FRET as a function of acceptor concentration. So here you can already see it looks something like a binding curve, and that's exactly what we are after. We need to get the whole binding curve to be able to determine the association constant. And then for each area of a cell, we also plot the donor concentration versus the acceptor concentration. So here you can appreciate how the concentration can vary. Like, for instance, in that cell and in that cell, the donor to acceptor ratios would be very different, but we take this into account. Another thing, you should appreciate the large range of concentration that we need to achieve in our experiments to be able to get a binding curve. And these are all the data you need to go into analysis. So we ask, what is the best model that describes the data? Is it a monomer only? Is it a dimer only? Is it a dimer-monomer equilibrium? Is it trimer, and so on and so forth? So then it turns out that the best fit is achieved when you consider the dimer model. So we know this because we look at the mean square error as a function of oligomer order. And furthermore, we see that the model that describes the data is one that monomers and dimers are in equilibrium. So the data are described by a dimerization curve shown here with the solid line. So this is really the law of mass action. The more receptors I have, the more dimers I have, the higher dimeric fraction I have. So the solid line is the fit, and the data are represented with these solid circles where I've also binned the data in small concentration intervals. So now the question is this really important? So the way to evaluate the importance of dimerization is to look at endogenous physiological receptor concentrations. So in lung cancer live, FA2 expression is about 600 receptors per micron square. So this is this dotted line. So we see that in lung cancer cells, 70% of the receptors are actually dimeric. So this is highly relevant in biological context. What we measure is highly relevant in biological context. But now, is it really important for function, and does it really play a role in oncogenic signaling? So the way to figure this out is you need to somehow destabilize this dimer. So this can be done via mutagenesis. So we were very lucky. We were able to destabilize the dimer by mutating amino acids here in the second domain. So these are the results. As you can see, dimerization goes down. Data are shown in red. For the same concentration, you have higher dimeric fraction for the wild type, lower dimeric fraction for the mutant. So now what we were able to create, a dimerization deficient mutant, and now we have the wild type. So now we can go into functional testing. So what is the function that interests us? Well, that would be migration, because as I already mentioned, migration correlates with metastasis and with invasiveness. So what we do is we measure migration of cells that express either the wild type or the dimerization deficient mutant. And what we see is that the mutant gives rise to way more motile cells. So there is this correlation where high stability of the dimer correlates with low migration, and reduced stability of the dimer is red correlates with increased migration.
Mm-hmm.
So what this led us to think about is that perhaps the proteoborigenic signaling is mediated by the monomer.
Okay.
And that then there is the idea then when dimerization occurs, it is anti-tumorigenic. So in lung cancer lines, 35% of the receptors would be monomeric. So according to this analysis, it may be that the proteomerigenic activity is carried by the monomers. So we have an idea how to test this, and in fact, this has relevance to cancer therapies. Then if we are able to stabilize the dimer additionally, then we'll be able to inhibit proteomerigenic signaling. So this slide illustrates the idea. If you can shift the equilibrium towards the dimer, then proteomerigenic signaling will be inhibited. So we have already done some preliminary experiments with a small peptide that was discovered in the lab of our collaborator, Elena Pasquale. So it was already known that this peptide can inhibit proteomerigenic signaling, but the mechanism was unclear. So what we shown that indeed this peptide stabilizes the dimer. So we believe this happens via an allosteric mechanism where the interface gets modified when the peptide binds here in the binding sites. So we are still working on this project. But what I would like to emphasize for this talk is that here a new biology was discovered. We discovered that unliganded dimerization happens in under physiological condition, and we discovered that it has anti-tumorigenic activity. And these discoveries were possible because of the precise biophysical measurements made. So this is a case where technology allows you to make very precise measurements. And when you do these measurements, sometimes you will even discover new biology. And in fact, we have multiple examples in our recent work where this is happening. So I think it has been very difficult to make quantitative measurements of interactions in the plasma membrane, but they can be really revealing of mechanism. So I would like to say, if you're interested, we'll be happy to talk to you. So with this, actually, I would like to finish. I would like to thank my team and, the Fred people on top. It has been really a pleasure to work with them, and they are the people that move us forward. It is not me. So credit goes to them exclusively. So with this, I would like to finish.
Can you please unshare the slide? Thank you.
Okay.
Yeah. Thank you very much. It's a pretty nice talk, so I really enjoyed it. I have a couple of questions.
Okay.
So have you studied any tetramerization or anything with this, or like any other receptors?
Okay, so we work predominantly with receptor tyrosine kinases, and they tend to dimerize. However, we have discovered that sometimes a single mutation in these huge receptors can lead to the formation of oligomers.
Yeah.
So we have certainly reported that it is quite interesting how this is happening. Unfortunately, we don't have structures of full-length receptors. There are exactly 0 structures. So we still don't know exactly what is happening on the single amino acid level. But to me, it's surprising how interesting it was to make an oligomer out of a dimer by a single amino acid mutation.
And like there is intracellular calcium also helps in the dimerization. There was a recent paper which shows that in science signaling that shows increase in intracellular calcium helps in the tetramerization of IP3 receptors. So like that helps in the secretion of pancreas and saliva, everything. So have you like tried anything similar, like increase in the intracellular? This is what we are talking about is extracellular currently, like ligand binding from outside the cells.
No, actually we haven't, but we do make measurements in both plasma membrane derived vesicles and cells. And sometimes we don't see big differences between the two. So we don't think that intracellular molecules are contributing significantly to what we measure. Also, keep in mind that what we do is we vary our concentrations over a very broad range. So when we get to very high concentrations, then let's say soluble factors, cytoplasmic protein may be just too few for the huge, overexpressed population of receptors. So we may not be seeing it because of this reason. So in fact, we are thinking about the next steps, and we would like to develop technologies now to measure interactions between soluble proteins and the membrane receptors under controlled conditions. So that would be the next set of projects, and that's what we'll be doing for the next few years, actually. That's the plan. I hope it works.
That would be interesting then. Like I have one more general question, like would you be interested in accepting postdocs in your lab? Like would it be okay if we can put your email address on your presentation and all those things? Would it be fine with you?
Certainly you can put my email address. I don't think we are looking at a postdoc right now because right now a few students are graduating and new ones are coming, so we already have problems with space at that very moment. But the students that are about to graduate will graduate, and there will be space then.
Okay. So that would be interesting, and to see like your future work. And thank you again for taking time and presenting your interest.
My pleasure. Thank you for having me on
Hangout. So certainly feel to contact us if you think that there is something new happening in your lab and you want to share it with people.
Absolutely.
Thank you again.
Bye-bye.
Bye.